You're watching CTOM TV. Knowledge is power.
This is a wonderful new technology. We're so happy to have this. We've been using it for what? A little over a year now, Michelle, is it?
Yeah, about a year and a half is when
Year and a half. Incredible technology. So this looks at the expression of the genes from a tumor sample once again, but it counts the amount of RNA molecules. And RNA are basically just the templates for the protein. And so basically it tells you how much of that gene product is being produced by the tumor cell. And it compares it to two samples of your normal tissues or of normal tissues, standardized normal tissues. And it says, okay, your ALK gene is overexpressed 200-fold. That means ALK inhibitors will benefit you. Or your RAS gene is, your KRAS gene is overexpressed, or your mTOR gene is overexpressed, or your tumor suppressor genes are underexpressed. So a mutation typically causes a gene to be overexpressed or turned on permanently. So a mutation changes the structure of a gene, so it can have different functions and have different functions that it was designed to do, and therefore they can be cancerous functions. And so tumor DNA sequencing looks at mutations. RNA expression testing just looks at the amount of that mutation. And this is really important because one of the things that happens in tumor DNA sequencing is you're going to get a bunch of mutations that are called variables of unknown significance. And these are mutations that happen in these cancer-causing genes, but they're unique to you and they've never been discovered before. And therefore, we don't know if they make the gene cancerous or not. We just know they're in your tumor. They could be benign mutations. They could be non-driver mutations, and we don't know. So they're just called variables of unknown significance. Well, the beauty of the RNA sequencing is it complements tumor DNA sequencing by telling us, yes, you have a variable of unknown significance in your ALK gene, but you also have 1,000 or 500-fold overexpression. So that's not a variable of unknown significance anymore. That's an activating mutation that is causing overactivation of your ALK oncogene. So it allows us to tell whether these VUSs are actually causing a problem to your genes or not. Additionally, the company we use has this artificial intelligence, and what they do is they look at all of the overexpressed genes in your tumor sample, and they say, okay, and they match it against a database of drugs. And this database basically, so every drug is going to have a beneficial, it's going to turn on or turn off a certain set of genes in your tumor. So a drug like a chemotherapy drug, like docetaxel, will typically, let's say it's gonna turn down 7 pathways that cause tumor growth, and maybe turn on 2 pathways that cause tumor growth in your tumor. So the overall balance is 7, and then there's 2 that it causes the tumor to grow in. So every drug is like that. Some drugs more than other, but the effect of each drug on your tumor is going to be unique to you because the expression of your genes and your mutations are unique to you. So in one person's cancer, docetaxel may turn down 7 tumor-causing genes and turn up 2. In another person's cancer, it may turn down 14 tumor-causing pathways and turn up 7. So this artificial intelligence scales the positive and negative effects on your specific tumor of a massive amount of genes or drugs in this database. And so then it picks the best 10 drugs for you, and it scales all the other drugs in this Excel file. So you have this massive Excel file, and you can say, oh, well, how's this drug going to work for me? Well, that's listed as number 167, by the computer. Whereas this drug over here that's for breast cancer is listed as number 2. And so there was a clinical trial that looked at this, and they found significant benefits with this approach. So artificial intelligence, computer-based technology. Now, the problem with that technology is the database it uses of drugs is well established, and new drugs come out literally every couple of days. So in order for that database to be upgraded, which it is not, it's still a little old, they have to go through a rigorous process to make sure this artificial intelligence is actually doing what it's supposed to. So what we do is we take their raw data and we look up these new drugs and do a by hand analysis after the computer has done what it's done. So computers are stupid. They only do what you tell them to do, and they're not going to know that there's a new drug for ALK overexpression. So if we know there's a new drug for ALK overexpression, and we see, you have high ALK expression, we're going to recommend an ALK inhibitor.
Thank you for watching Cetome TV. Subscribe below and stay informed.